Good morning, everyone. Today we are going to discuss about proteins. What are proteins? Proteins are polymers of amino acids, or we can also call them as the polypeptides. And these are basically made up of the following elements: carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, as well as there are few quantity of iron, iodine, copper, magnesium, as well as sometimes there is also the presence of iron, iodine, copper, magnesium. Sometimes there is also the presence of manganese. So here, this is how the polymers of amino. This is how proteins is made, and a basic composition of protein is in such a manner that 60 to 65% of a protein is composed of nitrogen, apart from which uh, 20 to 25% protein is composed of carbon, apart from which 10 to 14% is composed of oxygen, apart from which 7% of protein is composed of hydrogen, and 0.3% to 0.5% is composed of sulfur. Apart from which, this is the basic composition and how is actually proteins made up of. This is basically not the composition of protein, it is the composition of amino acids. And proteins, why these are important? Because these are the basic structure, they form the basic structure and function of uh, human body and other organisms, because 50% of the dry weight of cell is protein, as well as 15 to 18% of the protoplasm of cell, or the cell as a whole, is having protein. Apart from which, there is uh, formation of protein, which is done by peptide bond. What are the monomers of protein? These are amino acids. Amino acids are joined together by the peptide bonds to form the polypeptide chains or proteins. And when it was discovered by Berzelius in 1838. Oops, it's 1838. 1838. And then a scientist named Mulder came, who described the significance of protein. After which, let us describe the structure of amino acids now. Uh, Basically, in our universe, we are having different types of proteins. In the human body, there are 10,000 proteins, and all of these proteins are made up of different types of amino acids, which we are going to further discuss. So each protein is made up of around 3,000. If I'm not wrong, it's 3,000 or more than 3,000 amino acids constitute protein. Now that depends on what type of protein it is. So here, um, three types of protein, 3,000 or more amino acids are joined together to form the protein structure. And how many types of amino acids are there? There are 170 types of amino acids, out of which uh, 25 of these constitute the protein, out of which 25 constitute the protein, out of which 20 mainly constitute the protein. And uh, what are these 20 amino acids? These are present on the downward side. All of these are the 20 amino acids which constitute the human body. All of these are 20 amino acids. And here, uh, before discussing all of these, let us discuss the structure of amino acid. Amino acid is basically joined together. It is combined. It is having an alkaline or basic amine group, which is constant, as well as a constant carboxylic acidic group. carboxylic acidic group as well as amine basic group. Both of these are joined together to the central carbon, which is known as the asymmetrical carbon or alpha carbon. And apart from that, on the other side, we are having hydrogen, and this is R group, radical group, or we can also call it as alkyl group. This radical alkyl group is basically a variable group, which means it changes in the structure of amino acid, and that is the major uh, group which determines the chemical properties of amino acids when it comes to forming different kinds of amino acids. So there are 20 basically kinds of amino acid, and in all of these structures there is only a difference in the amine group. Oops, these are 20 amino acids, and in 20 amino acids there is only the difference in the radical group, while amine group and carboxylic group are remain constant, as well as the hydrogen group. This structure is constant, whereas this structure of the radical group is variable, which means it is changed due to which it determines the chemical properties of the amino acids. And simplest amino acid is glycine. It is basically having its simple formula as C over here. As you can see in the diagram, C is present over here. There is carboxylic acidic group, amine group, hydrogen group, and its alkyl group is hydrogen as well. Apart from which there is the second there is the second uh, simplest amino acid, which is alanine. Alanine is only having one alkyl group. Its alkyl group is basically methyl group. So here this is also having a simple structure, but this in this structure it is not having uh, a hydrogen, which means it is not having a hydrogen on the third end side. Now protein is this is these are all of the amino acids which are having different alkyl groups. For instance, serine is having hydroxyl group, and on the other hand side we are having threonine, methionine, and leucine, as well as there are different types of amino acids. Now we are not gonna discuss each and every one in detail. We are just gonna move on to the structure. And now, for suppose this is one amino acid which I have discussed over here. This is a single amino acid discussed over here. Now a similar amino acid is joined together with this amino acid. For suppose we have another amino acid which is present over here. This is one amino acid, this is another amino acid. Both of these are combined together. When both of these combine together, form they undergo dehydration, synthesis, condensation, and due to which one water molecule is released due to the liberation of hydrogen and hydroxyl ion. Hydrogen is obtained. Hydrogen atom is obtained. Hydrogen atom is obtained from the amine group, hydroxyl group, hydroxyl atom, hydroxyl group. Sorry, hydroxyl group is atom from the carboxylic acid, because amine group doesn't require hydrogen, carboxylic group doesn't require hydroxyl. Both are amine group is acid basic and carboxylic group is acidic, due to which both of these do not require the hydrogen and hydroxyl group respectively. Both of these combine together form H2O, due to which water is liberated and they collectively are joined together by causing to form a bond between them, which is known as peptide bond. And when both of these combine together they form dipeptide. And similarly, if there are three molecules, if there are three amino acids which are combining together, then that would be tripeptide. And accordingly, when there would be four, there would be tetrapeptide, and when there would be many amino acids joining together by the peptide bond, that would be polypeptide. So this is how it works. After which we are having some other terms, the types of protein structure. So here we are having different types of protein structure. First we are having the primary protein structure. What is a primary protein structure? It is the protein structure. Uh, it is a structure which is in the linear shape, which is in the linear chain or a straight linear chain, which consists of amino acids, and that is having those amino acids joined together by the disulfide bond. This is joined together by disulfide bond, and this type of structure, primary structure, is found in insulin. Secondly, we are having alpha helix beta pleated structure. Alpha helix structure is also present in the cell membranes alpha helix protein, which contains the alpha helix protein structure in actual. Secondary structure is basically the structure which is present. Uh, Secondary structure is a structure which is having the amino acids which are folded to each other. As you can see in the diagram, alpha helix and beta pleated are two types of secondary structures, which both of these are folded with each other. Both of these are having amino acids which are folded at different points due to hydrogen bonding. And how does it takes place? It takes place by hydrogen bonding between group of charged amino acids, due to which there is hydrogen bonding, and that hydrogen bonding leads to folding of the linear chain of protein, and then this leads to formation of the secondary structure. And this secondary structure is made up of disulfide bond and hydrogen bonding, and it is having alpha helix protein and beta pleated sheet. Alpha helix protein is a protein which is basically having the movement of alpha helix protein, which is just in the shape as it is mentioned in the diagram, that alpha helix protein uh, is a protein which is having its foldings in the form of a helix structure. On the other side, beta pleated is having its foldings linearly. There is first folding, then there is another hydrogen bonding, after which there is another chain of amino acids, then there is another folding, and so on. These are arranged in such structures. And this is the secondary structure, and secondary structure is found in which type of protein? It is found in fibrous protein, which we are going to further study classification. So here tertiary protein is a protein which contains both types. It is tertiary protein which contains alpha helix protein as well as beta pleated protein, and this is containing uh, more foldings of amino acids. This is having more foldings of amino acids, as which contains the alpha helix and beta pleated sheet. And these alpha helix and beta pleated secondary protein structure are joined together by ionic bond as well as disulfide bridges. First, there is in this type of structure there is disulfide bond, hydrogen bond, ionic bond, as well as disulfide bridges. Now, when it comes to quaternary protein, it is a quaternary protein which is made up of more than one type of globule. Now, this tertiary structure is known as globule. Now, if there are several tertiary structures, suppose this is one tertiary structure, one globule, there is another tertiary structure, another globule, there is third tertiary structure, which is another globule, and there's fourth tertiary structure, which means there are more than one chain of amino acids which are bonded together. More than one chain of amino acids bonded together collectively form a structure known as the quaternary structure of protein, and that structure is bonded together by hydrophobic interaction alongside disulfide bond, disulfide bridges, hydrogen bond, and ionic bond. And quaternary structure is found in the structure of hemoglobin, which we are going to study right now. Now let us study the significance of amino acids. Significance of amino acids was first described by F. Sanger. That F. Sanger said that amino acids are important, and these are important for every organism, and inside every organism's in body. Apart from which he described, he said that proteins are important, and he observed the significance of amino acids in the chain of insulin, and insulin is a proteinase enzyme. Oops, sorry, actually, now uh, sorry, so this is So insulin is basically a proteinous hormone released by the beta cells of pancreas, which was observed by F. Sanger, and then he realized that to describe the significance of amino acids, that amino acids sequence inside the structure of a protein is important, and that determines the functional role of the protein as a whole structure. So here, amino acid is a structure inside insulin and hemoglobin was observed by him. And what he observed was this: there were two chains of amino acids. First chain had 21 amino acids. Second chain had 30 amino acids inside the structure of insulin observed by F. Sanger. So he observed that there were bondings which were known as the disulfide bonds between the chain A. Let's suppose that it is chain A, this is chain B, and there was bonding between chain A and chain B known as disulfide bond. Now the seventh amino acid of A was joined with 7 amino acid of B, as it is mentioned on a diagram as well. Secondly, the 20th amino acid was joined with 19th amino acid of chain B. 20th amino acid of chain A joined with 19th amino acids of chain B. After which there is a bonding. There's an other bonding. Actually, there are two disulfide bonds, but there's an other internal disulfide bond within the chain A. That is from A6 to A11, which means 1, 2, 3, 4, 5, 6, 6 to 11, 7, 8, 9, 10, 11. So there's another internal disulfide bond within the amino acid chain A, which is known as the disulfide bond, which is between A6 and A11. And this was a structure which was determined by him. He found a structure apart from which we are having hemoglobin structure. Hemoglobin is a pigment which causes to transport oxygen in our body and carbon dioxide in our body to some extent as well. So here first we are having its two parts. First part is heme group, second part is a globular protein. Heme group consists of four pyrrole rings which consist of four iron atoms, apart from which we are going to further here discuss about the major structure, which is the globular protein. So second part is globular protein, which consists of four chains. Two of these are alpha chains, while two of them are beta chains. Alpha chains consists of 141 amino acids. Alpha chain consists of 141 amino acids. Beta chains consists of 146 amino acids, 141 and 146 amino acids. And here alpha one alpha chain is having 141 amino acids. As there are two alpha chains, there would be 282 amino acids. And similarly, we have a two beta chains, so it will become 292 amino acids. Both of these will combine together to form 574 amino acids inside the structure of hemoglobin globular proteins. Apart from which, there is also another protein which is known as histidine, which is present inside the structure of hemoglobin in order to stabilize the structure of globular proteins, which is known as histidine. Histidine is basically present in alpha as well as beta chains. In alpha chains, there are 10 histidines. In beta chains, there are 9 histidines. Apart from which, these are quaternary protein structure, and this quaternary proteins structure was previously discussed. So now, from this, there is also a disease which is caused due to the dislocation of amino acids or disturbance in the sequence of amino acids. So, F. Sanger also elucidated that if the sequence of amino acids inside a protein structure is disturbed, so the complete function of the protein is dislocated, complete function of protein is disintegrated, due to which the protein is unable to perform its major function or structural role. Here, there is a disease which is a genetic disorder known as sickle cell anemia. Sickle cell anemia. This is a condition in which the red blood cells become sickle and crescent due to point mutation in the DNA which produces the RBCs. Due to the point mutation in the DNA which produces RBCs, there's a point mutation. mutation in the single or more than one or single or few nucleotides. It is a mutation in single or few nucleotides due to which the it causes this point mutation, single or few change in single or few nucleotides in beta globulin protein, beta globulin uh, gene, beta global gene. I'm not sure about the spelling. Beta global gene, and uh, that produces a mutation that's in the structure of uh, beta chain. Inside the structure of beta chain of inside the structure of the beta chain of amino acid, it causes to produce a mutation in such a manner that the sixth amino acid of the beta chain is replaced by another amino acid. Sixth amino acid is in general, it is a glutamic acid. It is glutamic acid, but that glutamic acid is then replaced in the beta chain at the sixth position with valine, which is another type of amino acid due to which it causes to disintegrate its function, and it becomes unable to transport oxygen from the body, and it causes to lead to the condition of sickle cell anemia. So finally, we are having classification. Classification of protein is in such a manner that we are having two types of classification of protein. Proteins are classified into two types based on their shape and structure. First includes fibrous, second includes the fibrous proteins as well as the globular proteins. Fibrous proteins are proteins which are straight, which are these proteins are basically the secondary structures, and these fibrous proteins are secondary structures. And as you can see in the diagram, that is having a secondary structure in such a manner, there is undistorted alpha helix protein, supercoiled alpha helix protein, as well as left-handed coiled coil. So here this is it is both alpha helix as well as beta braided sheet of secondary protein structure. Apart from which, these proteins are having several characteristics. What kind of characteristics are they having? So basically, let me just write it down. Fibrous proteins. Fibrous proteins are basically elastic in nature. These are insoluble, as well as these are elastic, insoluble, and these can't be crystallized, non-crystalline, as well as these are spiral shaped. These are spiral shaped. Apart from which, uh, as these are non-crystalline spiral shaped, these perform structural role, such as we are having actin, myosin, as well as a spider silk web is also made up of fibrous protein. Apart from which, there's actin, myosin, and the proteins, the in, the proteins which are present inside the cell wall, the glycoproteins which are present inside the cell wall, which surface cell surface proteins, are also examples of this, as well as the other types of proteins such as collagen, as well as uh, fibroin protein, as well as there is another type of protein known as there's another type of protein as well. Keratin protein is there, collagen is there, actin is there, myosin is there, as well as there's another type of protein which is known as. So this is what we call as the fibrous structure. In globular structure is basically inelastic. It is inelastic, and that is soluble in water. It is crystalline in structure. sometimes it is crystalline, and that is basically, as the name suggests, it is globular. It is having ellipsoidal shape or oval shape, ellipsoidal or oval shape, and that is performing a functional role. For instance, we are having example of hemoglobin. The globular proteins of hemoglobin perform the functional role of transportation of oxygen, as well as when it comes to other proteins such as ovalbumin is a protein which helps in embryonic development of embryo during the embryonic stage of reproduction, after which uh, there is another type of protein such as we are having casein. Casein is a milk protein, and that also helps in the storage functions inside the body, helps in storage functions inside the body. Apart from which, there are other types of proteins such as we are having lots of examples such as proteins which include hydro. It includes proteins such as so I guess these are the proteins which I have discussed here, and further examples can be collected from different sources and Google sources. You can collect it from there. Hopefully, you enjoyed the video, and we'll meet again in